Aloha, everybody! In this lab, we are going to learn about all the pigments that make leaves and other parts of plants the beautiful colors that they are. Now, if you've ever been to the East Coast in the autumn time, you've seen a spectacular array of all sorts of different colors of plants, reds and pinks and purples and bluish colors and all sorts of colors. Where do those colors come from? How do the leaves change color? Well, the answer, which you may not have known, but now you can tell all your cool friends at parties, is that the leaves are already those colors. Naturally, the leaves are producing those different color pigments, which are really just proteins. They're producing those pigments so that they can absorb as much of the light spectrum as possible for them to undergo photosynthesis. Now, photosynthesis, as you might recall, is kind of like the opposite of cellular respiration in animals like us. In our cells, we undergo aerobic cellular respiration, where we breathe in oxygen and we eat food, and out of that food we extract the compound C6H12O6, better known as glucose, and that oxygen and glucose end up going into our mitochondria and undergoing chemical reactions to produce energy that we need, ATP, adenosine triphosphate, and we breathe out carbon dioxide. Well, where do we get our oxygen? Where do we get our food? Well, of course, we get it from plants, because plants, through the process of photosynthesis, can produce their own food. That's right. These guys are autotrophs. We're heterotrophs. They're autotrophs. Auto means self. They're self-feeders. They can produce their own food. Wouldn't that be nice if we could do that too? Well, they're going to take that sunlight and they're going to absorb it, and it comes in little packages called photons, and they have proteins in the thylakoid membranes of the thylakoid discs of the chloroplasts inside their plant cells. You remember chloroplasts? And these proteins absorb different wavelengths of light. So in the case of chlorophyll, there's a couple of different kinds of chlorophyll protein. That's the protein that makes leaves green. But during the fall, when the temperatures drop and the sunlight regime changes, these plants stop producing chlorophyll. In the East Coast, so I'm talking about the plants that the leaves turn colors in the fall. They stop producing the chlorophyll and suddenly unmask all the other pigments that produce the beautiful colors, reds and purples and yellows and oranges. And so what we're going to do today is we're going to extract the color pigments from the leaves of plants. And it's actually a very simple procedure, but it's very, very cool, and it makes a beautiful piece of art called a chromatogram. So we're going to see what this guy holds in store. Now, I've chosen a hibiscus plant because, well, I live in Hawaii and I had hibiscus in my yard. You can choose any plant you want. In fact, it works really well to choose things like spinach or kale. You can go have a field day at the grocery store or even go to your backyard. Try to pick a non-toxic plant to be on the safe side. But anyway, I picked hibiscus, so I'm interested in finding out what pigments and in what ratios, what kinds of pigments are in this leaf making it the color it is. So what you're going to need a pair of scissors. Of course, you need your plant leaves. You are going to need one of your 15 milliliter tubes with a cap. So make sure it has the cap. You're going to need a coin. Doesn't really matter which coin. I picked a nickel. You could pick a quarter, whatever you want. Depends on how high stakes you want to be. You're going to need your 6-inch ruler from your lab kit. Of course, we don't use the inches side. We're going to be using the metric side. So remember that between every 2 sets of numbers is 1 centimeter, and each of the little tiny ticks making that centimeter up is a millimeter. So there'll be 10 millimeters in a centimeter. What else are you going to need? You're going to need this little baggie from your lab kit, and it's labeled Photosynthesis Lab. Now this right here is chromatography paper. It's kind of like a kind of filter paper. It's already cut to the right dimensions for you. We'll just be cutting the ends to make a point. Now, notice I haven't taken this out of the bag. It's really important that you don't handle this with your bare hands because, well, one, we're working with acetone, but two, you have oils in your skin that will actually disrupt the flow of the pigments up the paper, so it'll affect your results. So you definitely want to wear gloves when you handle this. Of course, as usual, I'm out of gloves, so I'll make sure I hold it at the edges. You don't want to get your fingerprints on the paper. So I'm going to leave it in there for a sec. And it's just a very absorbent paper, and what it's going to do we're going to be putting extract from our leaves on here and putting it in a little bit of acetone, and that acetone is polar, and so it's going to be kind of sucking those pigments up the paper, and it's going to separate out those pigments based on their molecular size. So it's kind of like a sieve, like the larger pigments are going to travel slower, the smaller pigments are going to travel faster, and that will allow us to see the different colors and make a beautiful piece of artwork called a chromatogram. And then, of course, you'll be using your cell phones to take a picture of your chromatogram and put it into your paper and post it on the discussion board. Okay, here's the other important thing you'll need. Did I mention you need a pencil? No, I didn't mention this. You're going to need a pencil, not a pen. Pens will not work with this. You need a pencil, okay? And you need acetone, and AKA fingernail polish remover. So you can get this in the cosmetic section of your local drugstore. I got this at Long's Drug. Now notice this says pure acetone. That is better to use for this lab. The purer, the better. A lot of the acetones, the fingernail polish removers you get contain, they've added dyes to them, fragrances. They're more dilute, so they're not going to work quite as well. But this I got in the same section. It was $1.79, pure acetone. Now, depending on the state you live in, I live in Hawaii, they may require an ID check to purchase this, etc. But anyway, it's fingernail polish remover. Now, this stuff is very strong, and it is very volatile, which means that you don't want to sit there and sniff this stuff. It will not be good for you. So notice I have the window open, so you want to air it out and just keep it capped when you're not using it. It evaporates very quickly, and just don't breathe deeply into this thing. And of course, we're going to be wearing our goggles the whole time we do this. You should be wearing your lab gloves and of course your lab coat. So we're going to be wearing all those. All right, so we're ready to get started. This is really, really easy to do. I'm going to put this down here so you can see what it is that I am doing on the table. So again, you want to be wearing your gloves. All we're going to do is we're going to take out our chromatography paper. Now, because I don't have gloves on, which is shame on me. Do as I say, not as I do, of course. I'm going to go ahead and put on my goggles. And I'm going to just hold the very edges of this paper, especially since I don't have gloves. If you're wearing gloves, it's not quite as sensitive. But what we want to do is cut a nice little triangle at the bottom, and that will give a nice little tip for our acetone solvent to travel up. Okay, so what I'm going to do is I'm just going to cut the corners off on each side with the scissors. Okay, so this is kind of what I just created. So I just created this tip right here. See if you can see that. All right. And now I'm going to take my pencil and my ruler, and I'm going to go up. Let's see, make sure you're on the metric side. In this case, I'm going to go up 1.5 centimeters on here. So I'm just going to mark 1.5 centimeters from the tip. And then I'm going to use my ruler to just make a faint pencil line. Not too faint, you want to be able to see it. I'm going to make a pencil line across the bottom of this, just where I marked it at 1.5 centimeters. Okay? So now when I'm going to show this to you, what I did here. Can you guys see my pencil line? Let's see. Can you see my pencil line? Okay, so I made my pencil line right there, just going straight across. And now here's the fun part. Take a leaf, and you want to make sure that the non-shiny side is down on the paper. And we're going to use our coin to just start making strokes all along that pencil line, back and forth and back and forth and back and forth, and just get a really dark green pencil line here. So I'm going to put my leaf right about, it doesn't have to be exact, but you want it to be as thin and nice a line as possible. And you just use the coin to go back and forth. So after a single rubbing, here it is. But that's not dark enough yet. You want to do this about 10 times. You want to, over the same spot, you want to just get it really nice and dark. And again, the thinner, the better. You don't want it to be real too wide. So try to go over the same spot each time. You might have to play around with it. And depending what kind of plant you use, you may have to go more or less over this. So I'm just going to keep doing this for a little bit. And of course, if you're watching this as you're doing it, you can rub along with me. But again, the idea is the darker, the better. The darker green you have, the better your results are going to be. So just keep doing that until you get something nice and dark. And I think mine's pretty good right now. So I'm just going to do it a couple more times. Just make sure you're going over the same spot each time so you get a nice, rich, dark line. And again, mine's a little wide here. The thinner, the better. You just want to get that pigment all soaked into that one spot. Okay, mine's pretty good now. So here's where I'm at right now. So you can see my nice dark green line here. Again, mine's a little wide. If you can make yours a little thinner, it works fine, but it doesn't really matter. And now what we're going to do, we need our 15 milliliter tube here. I also, by the way, have a drinking glass to hold my tube. And I also have one of my transfer pipettes that I'm going to use to put some acetone in here. Now what we're going to be doing with this is we want to, again I'm trying to grab this guy without touching the thing. It's a little bit tricky. Okay, so what you're going to want to do is we're going to be putting our filter paper, our chromatography paper, into the tube, and you want to have some acetone in the bottom of the tube, but you don't want that acetone to come up to your green yet. You just want it to be enough so that this is dipped into the acetone. That acetone will be pulled up automatically. So what I'm going to do is I'm just going to put a little acetone in the bottom using the transfer pipette. So you can kind of measure it with your paper. I really just need about that much acetone, so it doesn't even reach the first 2 milliliter mark on the thing. So I'm just going to put it in the bottom. And again, you want to make sure you're ventilated. I'm going to put my hair back. You want to make sure that you're not breathing this stuff in. And you want to wear gloves, goggles, lab coat for this. Okay, so I'm going to take my transfer pipette. I'm just going to scoop up a little. Go ahead and put it all the way down to the bottom so you can see how much you're doing. And that's enough. So it didn't take much at all. Of course, put the cap back on your acetone. Okay, so I put the cap back on my acetone. When I'm not directly working with it, put the cap on your thing, because this stuff is very volatile. It evaporates really fast. So now I'm going to take the cap off. Again, you should be using a gloved hand. And there's some little thing on there. And just very gently stick this in there, and it should fit pretty good. You may need to take your pencil to kind of curve it around a little bit to get it in there properly. So mine is kind of not cooperating in that sense. So you want to just kind of curve it around. It should just stand kind of on its own into the tube. Okay, and then you just want to make sure the bottom is sticking into the acetone. So my acetone line is right there. And all right, now once you get it in, hurry up, cap it, and stick it in the glass to hold it up. And it's going to take probably about 15 to 20 minutes to do. Now what you're going to notice here is that this line, the solvent line, which is the acetone, it's the wet part, it looks wet. So watch it going. What you want to do is cut this thing off before it reaches the very top, but not too far, or not too soon, because you want to allow enough time for the pigments to separate, but you also don't want the solvent running off the end of the paper because it could actually pull some of the pigments off the paper and you won't see them. So you want to make sure that you're keeping an eye on this, and this is traveling pretty quickly. So let me see if you can see what I'm doing here. So my solvent line's already right here. So it's traveling quick. I mean, it's like a race. And just watch it. So watch the solvent line. You want to cut it off when it's about maybe 2 centimeters or so from the top. That should be enough to allow these pigments to separate. So, you know, it'll take a few minutes, maybe 10, 15 minutes. Go ahead and watch it. Don't leave it alone because you don't want the stuff running off. And then I'm going to pause this, and once mine gets towards the end, I'm going to come back and show you what to do next. So, okay, it's actually been about 15 minutes, and you can see that my solvent line is pretty close to the top, so I got to be quick and take this out. So I'm going to take it out. Again, have on your PPE, and I'm actually going to use the back of the pencil, the eraser, to pull this guy out without hopefully ripping it. You can hold it at the sides. And now we got to work quick, so we've taken it out of there. We need to get our pencil, and we're going to draw a line right at the top of the solvent, so how far the liquid moved. So I'm going to put it down on the table. and I'm going to draw a pencil line right at the top. So you got to do that quick before the stuff dries up and you won't be able to see it. Oh, this is beautiful. Now we're going to want this thing to dry a little bit. But you can see all these beautiful colors, and it's a little tricky. We're going to see if we can identify the different pigments in here. Now I'm sure my camera is not doing justice to this thing, but hopefully yours does. So what we're going to do is try to identify chlorophyll A, chlorophyll B, and xanthophyll at a minimum. You may have others. Xanthophyll is a yellow pigment, and it usually comes very thin. I can see a little bit of mine here. I don't know if you will be able to see it, but it goes right near the top of the solvent line. So mine is right there. So I'm going to actually take a pencil and draw a line where my xanthophyll is. I'm going to kind of outline it. And then you should be able to see, if your plant has both chlorophylls, you should be able to see a difference in two colors of green, chlorophyll A and chlorophyll B. So I've outlined my xanthophyll there. It's the yellow pigment. So I don't know if you can see where my xanthophyll was right there. And now I'm going to take a close look and see where I can see two different colors of green. And I can definitely see the difference here. So I'm going to see if I can label these. Chlorophyll A, chlorophyll B, and this is xanthophyll. Okay, so I'm abbreviating these, but let's see. So on mine, I don't know if you guys can see this. So the upper part is the chlorophyll A. The bottom part, which is slightly kind of darker brownish, more greenish brownish color, is chlorophyll B. And then, of course, we said the very top is xanthophyll. So so I'm going to show you a picture. I'm going to actually, in the lab report, I'm going to put a picture of a chromatography paper I've gotten from the internet that actually they had a very nice chlorophyll A, chlorophyll B, and xanthophyll from their particular plant. I use hibiscus, so it's not quite as strong for some of these. And again, I don't really know how much you can see of what I did in the camera. So I'm going to post that for you guys so you can use that as a model. But at least this showed you the techniques you do to do your pigment extraction. And again, the ones, the more you see of a particular pigment, the more of that was in the leaf. So you can see that my hibiscus actually had quite a lot of chlorophyll B in it. Very little xanthophyll and not as much chlorophyll A as chlorophyll B. So anyway, oh, and then almost forgot the analysis. So we have to actually create what's called an RF value. We're going to calculate an RF value. It's a relative measurement of these particular pigments, and we're going to do that for each pigment. So what we're going to do is find our line of where we actually drew initially, you know, where we put our pigment. So mine was right here, right? And we're going to use our ruler, and we're going to measure the distance that the solvent traveled. So we're going to use our millimeters, or you could use centimeters. So from the line where you initially drew to the top of where the solvent migrated. That is the distance the solvent, the acetone, traveled. And then for each of the pigments, you're going to measure to the top of that pigment. So from the line that you initially drew to the top of the pigment. So in this case, my chlorophyll B is 5.2 centimeters that it traveled. And then what we're going to do is divide the distance each of the pigments traveled divided by the distance the solvent traveled. And that's our RF value. So in your handout, you'll see a little table, and for each one of those pigments, if your plant had that pigment, you're going to calculate the RF value for that. And then there's some questions to answer. And don't forget to use your cell phone. Take a picture of your chromatography paper, and that will go into your lab handout as well as on the discussion board. And make sure you're putting this on some paper and using a pen or a pencil to label each of the pigments there, all right, with their RF values. So here is a picture of the final product. This one I pulled off the internet because it was done really nicely. They were probably using a slightly different solvent, maybe mixing acetone with petroleum ether or something that we don't want to do in our house. But just to show you kind of what the ideal results are going to be. Now, we're kind of doing a rough and dirty version of this. But here is where your loading line is. So that's where your pencil line would have been, where we did the rubbing. Here is their chlorophyll B, and so you can see how it's kind of a more greenish brown color. So you can use your pencil to trace around that, and you probably just actually measure from for your RF value for the distance the chlorophyll B pigment traveled. You measure from the loading line there right here to the top of the most representative point of chlorophyll B. So I'd probably measure it to there, divided by the distance from the loading line to the top of the solvent, which in this case is way up there. So, you know, how far the acetone traveled. So the distance chlorophyll B traveled divided by the distance the acetone traveled would give you your RF value for chlorophyll B. For chlorophyll A, I'd measure from here to here. So this is chlorophyll A right here. You can see it's a more greenish, deeper greenish color. So that divided by the distance the acetone traveled would give me my RF value for chlorophyll A. And finally, from here to there would be the distance the xanthophyll traveled. So this yellow pigment right here is your xanthophyll, means yellow pigment. Notice up here is carotene. So carotene gives plants kind of an orange color. You may have carotene, you may not. If there's a big distance between the top of your solvent line and the xanthophyll, you know that that's carotene. In the case of my hibiscus, I don't believe I saw any carotene. I think it was just xanthophyll. So anyway, but all the pigments you do have, figure out their RF value, then have fun.